Oh, sorry, I think they were calling me because I wasn't audible. Oops. I have this list of things to do when I start. I just didn't turn on my volume. Okay. So now I'm audible. Let me start from the beginning. I was telling you that I know you guys are in the Indian Standard Time, and I
Oh,
didn't
sorry,
know
I
you
think they were calling me because I wasn't audible. Oops.
So
I have this list of things to do when I start. I just didn't turn on my volume. Okay. So now I'm audible. Let me start from the beginning. Um, I was telling you that.
Yeah, I was telling you that we did germline mutation. If I didn't do germline, variant calling without talking about cancer, that wouldn't be complete. So, cancer variant finding is an absolute important area of research and area of actual application where patients are getting benefited by the drugs. So, in any event, um, in any event, so I'm going to go back to the slide where I left. I'm going to move to this is the slide I was in. We were talking about somatic mutation. So, sorry about my voice in the beginning of the lecture. So, in tumor mutation, the difficulties we have is mutations tissues are often contaminated in normal. Sometimes even 80% of the cells in the tumor tissue that you get are normal tissues actually from the same tissue but normal cells. So it becomes sensitivity becomes an important issue in finding somatic mutation. So what do I mean by that? I want to go back to germline mutation. Remember, germline mutations are in every cell in your body. So, and then even if you have gotten one mutation on your mother's arm or father's arm, what happens is 50% of the reads that are randomly sequenced fragments from your DNA, genomic DNA, will have the mutation. So when you do 30x coverage, what it means is at least 15 reads. It won't be exact, okay, because 30x coverage, as I told you in the beginning, it's not uniform, okay, because of the bias in sequencing, because of the bias in PCR, it's not uniform usually. So what happens is 30x coverage, you can almost expect 10 to 15 reads will carry the mutation, germline mutation. Somatic mutation. Somatic mutation also it can be in one arm of the DNA in the diploid cells. Perhaps every tumor cell has that mutation, but in the sample, if you only have tumor cells in your sample, then yeah, 50% of the reads will have the mutation. But it is never possible to get a sample like that. So what happens is now, although the tumor cells are 50% mutated, one arm mutated, because the normal cell is not mutated in either arm, there is a dilution effect when you're finding the variant. That's an issue here. So depending on um, the doctor who does the biopsy, how clean his hands are with respect to getting the tumor tissue sample. Is it slipping and he's taking it from the side, or is he taking from the center of the tissue? Pathologists can tell you what percentage are contaminated with normal tissue. So, in any event, you're not going to go keep poking the patient again and again. As bioinformatician, we have to make sure we give them the best results. Okay, so that's an issue. Sometimes doctors want to hear, even when the tissue sample has only 5% cancer cell, 95% normal cells, they want to find the mutation because it's going to help the patient. So how do we get them that? 30x coverage will not cover it. Even a single read will not have it. So even if you did 100x coverage, if you are with me, think about it. Even if we do 100x coverage, okay, if 5% of the cells are only tumor, because it's only there in the 50% of the thing, it's less than 2.5 reads will have the mutation. So you can be sure whether it's a mutation or a sequencing error or whatever. So you have no confidence when you do even 100x coverage. Of course, 100x coverage of human genome, it costs a lot, right? 100x coverage of human genome is 300 GB of data. That's crazy amount of data. That's one run of Illumina machine that we have upstairs, more than one run of Illumina machine that we have upstairs, and each run, each flow cell costs 12 lakhs. Okay, that is what we're talking about. So it's not even possible. So what do we do? Today we are going to talk about what is targeted sequencing. This is what we're going to focus today. Why am my pen is not writing? Hold on 1 second. Let me pick the right pen. Okay. I did get it. Okay. So targeted sequencing, what we're talking about. By doing targeted sequencing, we can get 1,000 or 10,000x coverage for really cost effective, because cancer patients don't have that kind of money. And this is actually a patient service, so you want to make sure that you give them what they want for the very lowest cost possible. So sometimes you give them 10,000x coverage and only screen couple of genes. Sometimes you can get 1,000x and cover maybe the entire exome. So what do you want to target is a question of how much money it costs, what the patient is willing to pay, and all those things comes into the picture, not just a science of variant calling. Okay. So next slide will tell you there are technologies. I'm going to not go into very much detail in technology because during the semester course, and Vibha ma'am is going to do details of all the techniques and all the methods that are used in sequencing from the days of Maxim Gilbert to today's NGS, PacBio, and Nanopore, and all that kind of thing. That will be fun to hear. But I just want to leave you with some sense of what are the technologies people develop for variant calling, so that we can call a variant and we can be useful. Okay. So you can do selected regions and sequence it. So yeah, how do you select regions? One of the ways to select region, which is something we have done for the TIGS project, is amplicon sequencing. In amplicon sequencing, what we do is, suppose you want to know what are the variants within a particular region of the genome, you can PCR amplify that, or you can PCR amplify several such regions and then send the PCR amplicons for sequencing without even doing the gel or anything. Just send it out for sequencing. It will sequence everything. It will sequence the real amplicon, false amplicon, everything. And we know we can find the real amplicon from next generation sequencing data. But the problem with this is PCR is good, but how many regions are you going to PCR? Can you do maybe 6 regions, maybe 10 regions? And how many samples are you going to do? Because every sample you have to PCR amplify it. So if you have 10 samples and you have 10 locus for where you want to look for variants, then 10 times 10 you have to do 100 PCR reaction. Okay, so it doesn't scale very well. Okay, so now you're saying, I don't know which regions I'm interested in, but I just want coding regions. I just want coding regions. I just want exome sequencing, all the exons, right? Then are you going to PCR amplify every exon? That's not possible, right? Because you may not even know where the exons are. And for every short exon, the PCR primers will be in the introns. It will be complicated. You can't do it. So there are technology that has been developed to sequence just the exons of human, like it's called exome sequencing. And how do they do that? Okay. So I told you yesterday about the GTF file, correct? All the exons of human from human genome that are translated is known. It is there in a GTF file. So one of the techniques that let me see what I have in the next slide, so I'll come back to it. Yeah, okay. So in this slide, I'm showing that there are many ways people have chosen to do restrict the regions they're going to sequence. What do I mean by that? You know, when you're finding variants, actually don't think of variant calling and reference genome and everything. What we are really trying to do is we have 3 billion positions in the human genome, going from left to right. Okay, 3 billion positions. You're going to interrogate when you're saying that I want to variant call. What you're saying is I'm going to interrogate each one of that 3 billion position to see whether a given genome has a different variant or not. Okay, so when I'm saying targeted sequencing, I'm saying I don't want to know the 3 billion position. I don't care intergenic position. I don't care intronic position. In exome sequencing, I only care if it is from a coding region or not. So now you're only going to interrogate variation within the coding region. So what you need to have is a given sample, you should be able to just get all the exons and not the rest of the DNA. For that, there are technologies. There are two companies. I know that Nimblegen also had a technology for that, but I'm not putting it here because most often people are using the Illumina technology to amplify the hotspot or the region of interest. Agilent has even, I like Agilent actually. Agilent Technologies is a much smarter technology for picking up regions of interest. And they also give you customizable things. So you say that I just want cancer genes. I don't even want all the genes. You can customize it. They have something called ERA. Okay. I have done this. ERA, it's an interactive site where you can upload say 20 genes you're interested in interrogating. Again, I'm using the word interrogating because out of the 3 billion position, you only want those positions that are coding for the 20 genes you're interested. So you're just going to send in this particular website. If you have an account, you can log in and you can list the gene names, like the standard gene names. If you put those gene names, because it's human, if it's from human, they already know where it's coded, which sites they have to interrogate. They will create what is called a sure select kit. Sure select kit, what it does is they are actually using the old microarray technology to do this now because they were one of the first pioneer in synthesizing oligonucleotide right on a glass slide. So what they're doing is they know the exon sequencing, right? Because in the human genome, you know all the exons, you know all the exon sequences. The GTF file provides the regions for that. So you take those exons, you actually put 200 bases of each exon, you synthesize it right on the microarray. Okay, and then the microarray, base of the microarray where the oligonucleotides are attached can be clipped. They do one enzyme reaction, all these 200 bases that are sticking on top of the microarray, they come in a solution. They're going to send you that solution. Okay, that is the sure select kit. Then it may have a tag, it may not have a tag, but that solution comes to you when you order a 20 gene panel, and then you put your DNA in there, and then only select for double stranded DNAs. And all the single stranded DNA which did not have a complementarity in the 200 base that they synthesized and they sent will get washed off, and then you separate, denature that and get the genomic DNA that only bound to those 200. That is sure select technology. It's beautiful, really. Okay, so they already sell you the exome kit, exome sequencing kit. Okay, so when you order, they send it to you. They are not cheap, $1,600 or something, but you can do 10 samples in that. Okay, so I know upstairs people do this very often. Our students have done that in the past, and you will know, eventually, you will learn how to create a exome library. Okay, and similar to exome library, as I said, the same technology they will use to make custom. So if you have, like, for example, the customized thing, one of the example I have used it before is, what do you call it? Cancer array. Okay, cancer. There are 409 genes that are listed in the Broad Institute website as consensus cancer genes. You want to interrogate all those 409 genes, you can order Sure Select Custom Array, and you say that, okay, I have interrogated all the genes implicated in cancer so far. Okay. In Illumina, they have gone a little further. Okay. In Illumina, what they have done is they have something called TrueSeq Tumor. TrueSeq Custom also they have, which I have not used, but probably very similar to what Sure Select Custom is. In TrueSeq Tumor, let's think about that. This is really very interesting, and in fact, Strand Genome, Strand Sciences, Strand Life Sciences in Bangalore, they use this often. This interrogates only those regions of the genome which are hotspot cancer mutation. How did they find the hotspot mutation? I will show you in few slides that follows. Okay. So they have said that forget cancer genes, forget everything, because we are not in a discovery mode. We really want to help patients. So they said we are just going to reduce the cost of sequencing so much, by only interrogating very limited region of the. So TrueSeq Tumor 26, it only has 26 hotspot it is interrogating. Then you have TrueSeq Tumor 170, which interrogates 170 hotspot region, and there is 15. The smaller the number of sites you're interrogating, the lower the cost is, and so you can offer it. And that's okay, because still, after all these, we are able to help only 10% of the cancer patient. We have long ways to go, and I'm hoping young students like you can contribute to how we can help more and more cancer patients. How do we do this better? Okay, so that's a tragedy of it. But so people don't want to spend a whole lot of money. Okay, so hotspot. How did they find the hotspot? I'll tell you about it. What the hotspot does is actionable. Actionable is a word you will hear all the time. What does it mean? Hotspot mutation may be diagnostics. You can say, oh yeah, you have cancer and everything. But people already know they have cancer. Why would they want to get this hotspot sequencing done? Because they want to know whether a given drug will work or it won't work. Okay, because they don't have much time in their life. They really need to get on the right drug right away. So those are called actionable mutation. And actionable mutation right now are very limited. There are kind of 10 to 20 drugs that the treatment will depend on a given mutation. So it's a very small amount of thing that's actually clinically useful variant. So that's how humbling it is. In spite of so much genomics we have done, it's very humbling that we only can do so much with respect to cancer. But good news is we are able to help. We are able to help 10% of the cancer patient, which is good. So in the next era, which is exome, so exome capture is a very similar. In next era, what they do is they don't cleave off the 200 base exon sequences, complementary sequences. They actually have it on the microarray, and they will come with the DNA of an individual, hybridize it on the microarray, and whatever binds, they will wash it off. Okay, and sequence the only thing that was bound and washed, not the free ones. Okay, so things like that, pretty simple molecular biology tools, but they have done a lot of optimization to make sure you cover the entire exome. You don't lose it. That's where the technology and optimization went. Hats off to them. They do a very good job. Okay. So, okay. Now I'm going to go. Where did they get the hotspot? Okay, everybody is getting to the understanding me. I'm hoping that there are two reason, or even more than two reasons for doing target sequencing. The first reason, to keep the cost low. Okay, and focus on the region of interest so that you can keep the cost low and find the variants with very high sensitivity, because now when you do 100x sequencing like 30 GB, then you got ridiculously high coverage. Correct? You're getting me, right? Let me go back to the coverage thing which I did not mention. Okay. Here, for example, if I'm interrogating the whole genome sequence, and a sequence say 30 GB of data. I've generated 30 GB of data. One lane of the HiSeq 2500 we have upstairs can give you 30 GB. I told them to sequence one lane. Okay, because that's all I can afford. Okay. Now, if I didn't do the whole genome, if we did the whole genome, what X coverage will this be? The coverage I'll get at each position that I'm interrogating. Remember, I'm interrogating 3 billion position. So this divided by 3, I only have 10x coverage. But for the same amount of data, if I'm only interrogating 30 million position, why I'm saying 30 million? Because our coding regions are only 30 million. Okay, very 1 or 1.5% of our genome are really coding. Okay, so 30 million position. Even if I'm interrogating every exon, every position that codes for an exon, I'm interrogating only 30 million position. So if I create the same kind of 30 GB of data, what is the coverage I'm going to get? 30 GB divided by 30 MB equal to 1000x. So for the same money, in every position, I got 1000 reads. So even if I have only 10% of the tumor tissue in the sample, I will still get 100 reads. That is evidence for a mutation, that's cancer mutation. You're getting it. So that is why targeted sequencing is so popular in cancer. Nobody does whole genome sequencing in cancer. Okay, so because it's not viable, you cannot find it. Okay, so now going to how did Illumina get TrueSeq, right? So I know these slides may be a little hazy, but I think what is happening here, I'm going to draw it. From here to here, there is a small, this is an exon, okay? I am not putting it big enough. On the right, I have magnified it. This exon is really long. But the whole exon, because there are so many mutations in cosmic database, I had to really shrink to tell you how many mutations in this exon they have found. Okay? This is an exon from EGF receptor, exon 20. There is a mutation in exon 20 called T790, which mutates into a methionine. Threonine to methionine, okay, mutation, which is deleterious. Why is it deleterious? You can go and see whether the threonine is really conserved. I've circled it here. You know what I'm going to, you can go to Google and search for T790 EGFR mutation. You will get the coordinates in HG19. That's what I did. And when I type in that coordinate, I get this region. Okay, this entire exon 20. Okay, so the region I'm amplifying here, you don't know, these are all not at the same position. Okay, although it looks like a straight line. Do you see a red straight line here? That kind of thing. Yeah, so they are not same mutation at the same side. This is a big exon. Okay, but this region, if you look at it in the UCSC browser, it's in exact same position. Okay, when I amplify this region, okay, this is what I see. oh, this is the one when I'm saying this portion is jagged, this is how it is. These are different positions, okay? This is so compressed that you see it in line. So this position has a mutation, this position has a mutation. This is a single nucleotide. Do you see how many evidence of it is there in cancer database, in cosmic database for this position? And then there are other mutations. So this is a hotspot. Right? This is a hotspot. Not only that, each line here is a sample, is a different sample, and majority of them are from lung cancer. So when you are there in UCSC browser, I may send you guys the coordinates of EGF receptor, exon 20 mutation. I will also send you the coordinates of other ones. Go there to UCSC browser and see it for yourself. Cosmic, why am I focusing on Cosmic database? Cosmic database has 1 million tumor samples sequenced. I think mostly exome, some could be genome, but these are all cancer samples. So in the vertical here, these are all cancer samples in which they have found this particular mutation. When you click on this mutation, click on any one of these mutations on UCSC browser, it will tell you which type of cancer it came from. Okay, so majority of cancer will be lung cancer. That's why EGF receptor exon 20 T790M mutation is a diagnostics first of lung cancer of a specific type, and I circled it here, you cannot see it, but remember I told you that in different species, is it threonine or is it not threonine? It's all over the place. It is threonine all the way in fish. All mammals, birds and fish, every one of them has a threonine at this site. Okay, it's all T here. Okay, you can see it. So you know that any mutation in this is going to be deleterious. And now we know it's cancer specific. Okay, so this is one way to find hotspot. So they didn't go one by one like that. They didn't know. In the hotspot mutation, what they did was like they went through the entire site, and they found out the peaks in cancer mutation. This is the P53 exon, where the mutation, you know that this is kind of going crazy in cancer, this particular site. So they found regions like this, and this will be a hotspot. This region is a hotspot. So in two-site tumor, they took 26 hotspot like that, and they put it. In the other one, they reduced the thing, and they reduced the depth they need for it, and they have taken other 150 of them. So that's how they found hotspot. The cosmic database was instrumental in finding. The cosmic database is not only from NGS era, even from microarray times, which we haven't talked about, and I think we will talk about microarray technology, because a lot of what we do with NGS today owes it to microarray technology. So we will do a little bit of microarray technology during the semester course. But even from the days of microarray technology, the mutation cosmic database has found mutation in cancer. So it is at least 20-25 years old. Okay. So in the next slide, I'm going to show. Look. So this is BRCA. BRCA1, everybody knows. BRCA1 or BRCA1. Every child knows BRCA these days, right? It's breast cancer associated gene. So this is a long gene, and when you go and press search for BRCA in 1819 UCSC browser, you will get the entire BRCA gene. The dark blues are exon. These are exons, and the lines are introns. Okay, you see that every exon has mutation in cancer. In different patients, these are all different patients. Okay, in some of them maybe 2 mutation in one patient. But you can always click on this red spot, on this red spot, or on this red spot, and get more information on which sample is it coming from, what kinds of cancer did they have. BRCA mutation, majority of them probably came from breast cancer, but BRCA1 is not an oncogene. That's another thing you need to know. BRCA1 is a tumor suppressor, and EGF receptor is an oncogene. TP53 is a tumor suppressor. Okay, so tumor suppressor and an oncogene have a very different kind of profile because tumor suppressor are often mutated like crazy in many normal people as well. So, and tumor suppressor, it is at least said that tumor suppressors mutation doesn't necessarily cause cancer, but as you accumulate mutation in tumor suppressor genes, and when suddenly an oncogene comes, tumor is not getting suppressed and it goes haywire. Okay, so why did I talk about EGF receptor? Because EGF receptor T970, that's the third bullet. Okay, third bullet in this slide. T790M, T790 something else also is causing mutation, but just the T70 position, 790 position, if it is positive, there are drugs like Tarceva, Golitrif, and Iressa that will work. If it is resistant, then you have, if it is resistant to this mutation, then Tagrisso will work. Okay, so these kind of thing, either the drug has been developed for this kind of mutation or by sheer use in clinical trial, they know that they respond to this drug better with this kind of mutation. So either way, they know the relationship. So when you find this mutation, a patient can actually be given the right kind of drug. The other mutation I'll tell you is BRAF V600E mutation. This is the classic mutation in cancer. Why it's classic, I'll tell you, because 30% of the melanoma tumor samples will have BRAF mutation exactly at V600, which becomes a glutamic acid. I did mention this yesterday as well. Okay, but in this particular slide, the point I'm trying to make is there are two drugs, Drabafenib and Trametinib, for this positive, BRAF V600 E positive for melanoma. So if they're positive, you give a different drug. If they're negative, you give a different drug. So these drugs, some of these drugs have been developed just in patients with this kind of mutation. Clinical trial was done in patients with this kind of mutation, so and they are benefiting because of that. And I know all of you are familiar with HER2 mutation, HER2 positive, right? If HER2 positive, meaning that you can treat the patient with Herceptin. If HER2 negative, you got to do something else. It won't work. And important thing here is not just the cost of sequencing. These drugs are also very expensive. So you don't want to give a wrong drug to a patient. So that's why this variant calling becomes really important, and people are spending money and carefully treat their patients without spending a lot of money when they're really in distress. I thought I had a slide for B. Okay, I think I showed you a slide for BRAF earlier. I will also send you guys the coordinates for BRAF. You need to go and look at it. And you need to look at, click on every cosmic mutation, and it's amazing how they found this mutation. You know, how many melanoma patients they've sequenced, and they found exactly every time it's BRAF V600E mutation they found. Okay, this kind of crazy. So in HER2, it's a slightly different story. It's called copy number variation. It's not mutation. So often, even in microbes, not just in human, even in microbes, in plants, perhaps I haven't heard about it, but in plants also CNV may be there. But Herceptin, what happens is, if a cell has to survive in a different environment, it wants to make a particular protein in large amounts, without changing the genetic genome, without changing the this is a live person, an adult person. It's not embryonic stage or anything. So what it does is, in tumor cells, it copies the same gene several times so that the gene is expressed in a very high quantity. So that's called copy number variation. So you will see in the genome next to each other, and there will be more copies of it when you're doing genome sequencing. Okay, so that's how HER2 came up, and copy number variation, not just SNPs and mutation we have to worry about. In cancer, we have to find a way to look into copy number variation, translocation, different things. But this whole summer camp, I'm only focusing on mutation because that in itself is a lot to digest. Okay. Again, as I pointed out, unfortunately, where am I now? Oh my God, I'm 45. Okay. So, unfortunately, only less than 10% of cancer patients are getting benefited by this genomic approach. We have long ways to go. So don't sit there and think, oh my God, everything is done. Now what am I going to do? You're going to solve the other 90%, solve the problem. So other 90% of the patients can also get benefited. Okay. Think about it. Read a lot. Be very creative in your thinking. Okay. Cancer patients need a lot of help, just as much as plant genomics people need a lot of help in crop breeding, and microbial people need a lot of help from bioinformatics into finding which kind of soil or which kind of gut microbiome is causing a disease or making people better. So this whole summer camp, we had variants, variants, variants. That's all we did. Don't think that that's all bioinformatics is, okay? It's not. This is only a very small part, and even in this small part, I did not go into details, believe it or not. I did not talk about algorithms, which you need to understand to be able to benefit from the results. You won't be able to interpret the data better if you did not know algorithm. So those are all things, it will come in the semester time. We talked about two variant, germline and somatic. Okay. So for variants, what did we do so far? We said we need reference genome to call variants. So we did what is reference genome, how to assemble it. We did why do we need it? Why do we need to assemble genome? And why do we need variants anyway? Right? To make sure we benefit from the variants, we need to annotate the reference genome. We need to know where the genes are. Is it changing the amino acid? Is it not changing the amino acid? Is it in the intergenic region? Is it just a silent mutation with no effect in the phenotype of a person or in the phenotype of a plant? So we need to annotate them. So we did talk about annotation. We talked about homology based and de novo annotation. You know the caveats in the annotation process. Yeah, I already know that some exons are over predicted in by Augustus in one of the genomes that we are interested in. So that we can always correct by doing transcriptome sequencing, which we haven't touched here. Okay, we have also done in the class in the lectures and in your hands-on. We have done how to call variants. I'm hoping all of you are familiar with mapping. Hope all of you are familiar with SAM tools, MPILUP. All of you are familiar with BET tools intersecting, and how do you interpret the variants. And then we talked about yesterday how to annotate the variants. How do you know a given variant is changing the amino acid or not changing the amino acid? So we talked about SNP effect database, how to create a database. If you have your own genome, you need to know how to create the SNP database. Okay, so now if for individual in cancer, you're looking for an individual to find out the variants. In population, we can also do population genetics using the variants from hundreds of individuals. And today we talked about how you can find variants to treat people. Okay, so that's it. Variant, variant, variant. That's what we have done. Okay, and what we haven't done. Oh my God. Okay, so you don't want to know about it, but we haven't done how to find structural variants. We haven't done how to find translocations, which are also a serious problem in cancer causing translocation is a serious problem. We need to find them. There's copy number variations. We haven't done transcriptome analysis. We haven't done splicing. We haven't done epigenomics, ChIP-seq, methyl-seq, which tells you epigenetic modification can change in cancer and in plant during development. Comparative genomics, that's a whole new ballgame. Two genomes from, say, Anne Stefanzi and Anne Gamby, how do you compare them? How do you know what got conserved, what changed, why they behave different? Some mosquitoes can only infect human, some mosquitoes can only go to cattle. They have some sensitive things. So their olfactory genes can change. So we can find out what's going on there. Metagenomics is soil samples. You can find out tens of thousands of microbes in a given spoonful of sample soil to find out why some soil are able to grow some plants with high yield and other soils are not. Metagenomics also covers gut metagenome. Gut metagenome is becoming a big part in human medicine as well because the composition of the gut microbiome is also known to cause much of our, diseases. So from diabetes to other things to cancer, it changes. Our metagenome changes, and then metabolomics, and then proteomics. So these are big area in bioinformatics. We have maybe covered one eighth of it just at the concept level. Okay, we haven't done details of it. So now let's go to quiz. This is the first time I'm going to quiz, so if it didn't work, it's okay. If it worked, then you just go ahead and I'm going to start. I'm going to go to the quiz scene. This is my quiz scene. I'm going to start the quiz. We did start the quiz. Something should pop up in this window, okay? Bear with me. Let me start it again. Working on it, it says, so it'll pop up. Sending to players, it says, so. You probably see on the screen what the rules of the game is. You should see. And you will have 20 seconds to answer each one of them, okay? So it's going to pop up anytime now. And it will automatically score. Waiting for the players to answer, it says. Okay, now I'm going to reveal answers. Oh, this is fun. Okay. Sending to players, it says. Show question score. Okay, do you see the scores there? Yeah, so, okay. You answer it right there by clicking. Maybe some people are answering on the chat. Okay, show total score. I have 10 of them, so it should end very quickly, okay? I do have 10 minutes, so it's fine. So next question. Everybody see their score? I'm going to go to the next question. It's going to pop up in 10-15 seconds. Okay, you can answer it now. I'm going to reveal the answer for question 2. The score is going to after this quiz is over, I'm going to check with you guys what you saw, because what you see is not what I see. So, okay. So I think you guys are going to guess. So I see what score. I have 8 more questions to go. Next question is popping up anytime. So it's waiting for all of you to answer. Okay. Now want to reveal the answers to you. No. Your score. Total score. I'm going to go next question now. I know someone is saying that some of us cannot see the quiz. I don't know. I got to work on it. This is the first time I'm trying this. Okay. So ask others how they are able to see it. Okay. So this question number 4, we're waiting for players to answer. Revealing answer now. Showing the scores. Show total score. Okay, 10 question does take 10 minutes. So that's okay. Next question. Okay. Now you know how the whole thing works. So I think you're getting a little easy with this whole thing too. So now you're getting question number 5, I think. It says waiting for players. Reveal the answer. Show question score. Total score. Next question. Revealing answer. Question score. Total. Next question. This is the seventh question, 3 more to go. Waiting for you guys to answer. Revealing the answer. Total score. Next question. I'm revealing the answer now. Showing the scores. Total score. Now it's showing 10 players, so I think some of you might have been able to join. I'm going to the next question. Showing question score now. score total? Last question, I think. I don't know whether it's last or last but one. Yeah, this is the 10th question coming now, and we're done. I'm pretty sure all of you got it right. Okay. So, but I will know at the end. I won't know what you scored. Okay, it's not going to tell me. It's for you. It's for you to see whether you're doing good or not. Okay. Okay, this is the final results, guys. Ready for it? There you go. Okay, I'm going to leave the screen for some time so you guys can get your final score. I don't see your name. It just tells me anonymous, anonymous. And three of you couldn't join. I think that's why it gives 0, 0. One person has a negative. But yeah, so it's good. I think it says end of game. I'm going to say end of summer camp. Okay, so I think this quiz worked out well. I'm going to end it now, and I'm going to go back to my camera mode to tell you, hey, I learned a lot about streaming. I don't know about you guys, whether you guys learned about variants or not. I'm pretty excited about this technology. Please, I'm going to give you 3, 4 minutes to tell on the chat how the whole thing went, the entire presentation, yeah, including the poll and the quiz and everything. Um, uh, uh, Out of 10, how much mark are you giving for this entire course? Okay, although I will see it, but be honest. Okay, be honest in terms of voice, in terms of my clarity, in terms of material that I material that I say have covered. Out of 10, just put the score on the chat box. Then also, I think there may be a place you can review it anonymously. So I will tell you where you have to go and value the score, value the entire course. Okay. So anyway, I'm going to wait and for 5 minutes to see how you guys rated this. It may not have anything to do with me. It can be the streaming you couldn't hear because the voice wasn't clear. I went too fast. And coverage course didn't cover what you thought it will cover. Anything. Okay. So just say it on the chat box. I'm going to go away. You just say it, it'll be stored. Okay? Okay, thank you guys for evaluating this. I know this was a lot of goofs were there. I think it'll improve. And the course material also, I had to shrink it a lot because I wanted to fit it in the summer camp. This is the end of the summer camp. I'm probably going to see the MSc OH students, the first class on July 15th. The very first class, it's mine. So I'll see you guys. Bye for now. Thank you.